to welcome back actually Lily Hazrati from University of Toronto and Sick Kids Hospital to moderate this talk. She presented earlier in the year and we're thrilled to have her here. And I'd ask you to please take this on and introduce them. Thank you so much.
Thank you very much, David. So we have Dr. Anne McKee today kicking off the first of the 2021 current topics in TBI webinar series. So I wanted to again thank you, David, Josh, and Adam for organizing the wonderful talks. It's been amazing to be exposed to them on a sort of a weekly basis. So it is with great pleasure that I get to introduce Dr. Anne McKee today. Dr. McKee is William Fairfield Warren Distinguished Professor of Neurology and Pathology at Boston University. She's the Director of Neuropathology for the VA Boston Hospital, the Director of the BU Chronic Traumatic Encephalopathy Center, and also Associate Director of the BU Alzheimer Disease Research Center. She's a board-certified neurologist and neuropathologist whose research has produced seminal advances in CTE and Alzheimer disease. And she has, looking at her CV, she has produced over 170 peer-reviewed publications so far, which is quite impressive. Dr. McKee created and directs the Unite Brain Bank. So as some of you know, it is the world's largest repository of brains from individuals exposed to traumatic brain injury and neuropathologically confirmed CTE. So Anne received her undergraduate degree at the University of Wisconsin and her medical degree from the Case Western Reserve School of Medicine. She was named more recently Bostonian of the Year by the Boston Globe, and was also named one of the 100 most influential people in the world by Time magazine. She's also the recipient of numerous prestigious awards, and I'm just going to name a few of them here. These include Henry Wisniewski Lifetime Achievement Award in Alzheimer's disease research, and the Samuel J. Heyman Service to America Medal, also called SAMI. Dr. McKee is also a member of the National Academy of Medicine in the United States. So I just can't wait to hear her talk. And I think your title, Anne, is CTE Status of the Literature.
Yes, thank you very much. Thank you, Adam and Josh, for inviting me, and Lily for introducing me. I'm looking at this title of mine, CTE Status of the Literature, and I'm wondering why I picked that. So clearly I can't do the status of the literature. I'm going to try to do the status of some of the literature, but there's actually been a very extensive amount of work done very recently in CTE. So I'm going to start out by telling you a little about myself, just because there's certain ironies in me studying CTE. One of them is that I was born in Appleton, Wisconsin, and that probably doesn't mean a lot to many of you, but it's where the visiting team stays when they play the Green Bay Packers. So when Tom Brady played Aaron Rodgers a few weeks ago, he stayed in my hometown. And I grew up in the 1950s and 60s, which was Super Bowl I and II. Of course, the Packers won that. My dad was a huge football player at Grinnell, and my brothers played football, and they had me playing football when I was whatever age that is too. And but I do think it's important that people realize that I came from a real football family where I really understood the value and the passion that goes into football. And I was actually quite an extraordinary football fan when I started this work. Of course, the work has changed things. I'm going to talk today about CTE. It's a neurodegenerative disease characterized by tau. It's certainly not a new disease. It was described clinically back in 1928 by Harrison Martland, who was a medical examiner, but he described the clinical syndrome that he said affected boxers or pugilists who stayed in the ring too long. The disease has gone by a number of different names over the years: punch drunk, dementia pugilistica, CTE. CTE was introduced about the middle of the last century, and that seems to be the term that has stuck, primarily because this is a disease that can affect many individuals exposed to many different types of head trauma. And then, of course, Bennett Amalu in 2005 and 2006, when he described CTE in the first former NFL players. I think that was the awakening of the world to CTE. And since that time, there's been an extraordinary explosion of information and scientific advance. I was very interested in those original articles by Amalu because I had seen changes in a boxer that had come into our university. He was in our Alzheimer's Disease Center. His name was Paul Pender, very well known in the New England region. He was twice world champion. He had twice fought Sugar Ray Robinson for the title. And he was evaluated by my colleagues in the Alzheimer's Center. And he was thought to have a case of Alzheimer's disease because he had retired from boxing in his early 30s and then was well and highly functioning until about his mid-50s, when he started developing behavioral problems, aggression. He started being violent with his family, and also developed a terrible memory and cognitive disorder. So the feeling was that he had Alzheimer's disease because it had been 3 decades since he had boxed. So I was very surprised when I did this autopsy. And the first reason was there was absolutely no beta amyloid in the brain, absolutely none. And so immediately, of course, Alzheimer's disease is off the table. And then when I did the tau immunostaining, even though I'd been studying tau-based diseases for many years, I'd been involved in the consensus conferences to establish the criteria to diagnose things like PSP and CBD, looking at post-encephalitic Parkinson's, looking at just a wide variety of tau-based disorders. And what struck me about this brain was I'd never seen this pattern of abnormality before. And it really, really fascinated me. It was extraordinary. First of all, it was florid Tau pathology, and then it had all these bizarre patterns, the way it curled around small blood vessels, it hugged the sides of vessels, it was patchy, it was irregular, it involved areas of the brain that generally are not involved. And so I became extremely interested in this disease and wanted to see and tried to see as many boxers' brains as I could. And then just by coincidence and really by luck, about 5 years later, I was approached by a man named Chris Nowinski, a former professional wrestler and Harvard football player. And he asked me if I'd be willing to look at the brains of football players. And of course, this was the collision, the intersection of just two enormous passions of mine. I'd been very interested in football, and then of course brain examination. So this was really just a gift to be offered the opportunity to look at the brains of football players. The first two individuals that came in were Tom McHale and John Grimsley. They were kind of strikingly similar. Both had been linemen in the NFL for 9 years. Both had retired from the NFL at 32. Both of them had developed some issues that were hard to describe by their wives, difficulty multitasking, some depression, some inattention, some memory problems, but very difficult to really pin down. And when they both died, Tom from an accidental drug overdose and John from an accidental gunshot wound, both families agreed to brain donation. And that was my opportunity to look at the first two football players. And their brains are shown in the middle of this panel. What was just astonishing to me was that the brains of these 45-year-old men, which by my standards, with a career in neurodegenerative disease, these men are extremely young. 45 is that the height of a person's life, and to see that they both had developed terrible neurodegeneration by that time, and that the neurodegenerative process that I saw in both of their brains was exactly like what I had seen in Paul Pender. No beta amyloid protein, this strange collection around blood vessels and in irregular parts of the brain, and really this floored me. I remember showing these cases to my brother, who was also a physician, and saying to him, I think we were, my brother's an ex-football player, and we both were just stunned. And our reaction was, we've got to let the NFL know. They'll want to do something about this. This is so horrific. Of course, my experience over the next decade was very different than that. And I won't get into my experience trying to bring this forward to sports organizations like the NFL, but you can imagine what it was. The other thing I did right when we first got those first two brains, remember that I had started an Alzheimer's disease brain bank. I had the Framingham Heart Study Brain Bank. I knew the power of brain banks to accelerate scientific advances by allowing tissue to be analyzed by multiple researchers at multiple centers. And so the first initiative of mine after getting these brains was to start a brain bank to investigate the long-term consequences of TBI. When I started out, it was a very small enterprise. I had two technicians. It was the two technicians and me. We did everything. We took the calls. We got the brains. We did every single part of the process. But over the years, it's expanded terrifically. It's been sort of almost a logistical nightmare at times to try to coordinate these brain donations, which come in 24 hours a day, 365 days a year. And it's brought a lot of attention to the brain bank, but I think it's really been the source of tremendous opportunity for scientific discovery. So what is CTE? It's a neurodegenerative disease. It's associated with exposures to repetitive head impacts. And repetitive head impacts includes symptomatic concussions, but as you all probably know, concussions have never been directly associated with the risk for CTE. What we've shown over and over, it's the cumulative repetitive head impacts, including the many asymptomatic subconcussive injuries that are experienced by contact and collision sport athletes. That's what we've found increases the risk for CTE. CTE has been described in all sorts of individuals, all sorts of sports players, but also people exposed to domestic violence, headbangers, as well as people exposed to blast. Right now, CTE can only be diagnosed with certainty at postmortem. It cannot be diagnosed with certainty during life. And if I had to pinpoint any goal that my team has, I think it would be to really focus on ways to diagnose CTE during life so we can really make some difference in the treatment and care of these individuals. Back in 2013, by that time, we had accumulated a series of 68 men with a history of exposure to RHI and evidence of CTE. And looking at those 68 different cases, and there was nothing magical about the 68 cases other than they represented a wide span of ages at death and a wide severity span. By the time I'd reached 68 cases, I felt that I had a fairly good handle on what I considered the real hallmarks of CTE pathologically. And so in the paper in 2013 in Brain, we described the diagnostic features of CTE, the hallmark lesions of CTE. And number one was the fact that the tau accumulates in neurofibrillary tangles and disordered neurites around small blood vessels, number one, which is a very distinctive formation and is unique to CTE. And it also accumulates at the crevices of the gray matter in what we call the depths of the sulci, where the gray matter dives in. That's where we see the preponderance of the early tau pathology. We also described in that paper a staging scheme of grading pathologic severity from 1 to 4. Again, there was nothing magic about the 4 stages other than that they appeared distinctive to me after looking at that number. The mildest individuals, the mildest cases seem to have just a few epicenters of tau pathology or focal CTE lesions, usually in the frontal cortex, sometimes in the temporal cortex, but just a few isolated cortical lesions. As the disease became more widespread and the lesions themselves expanded to different parts of the brain, we called that stage 2, affecting multiple lobes of the brain, and later we delineated it as 3 or more of these focal lesions in the cortex. Stage 3 was a more widespread disease. The individual CTE lesions were expanded. There was spread of the tau protein up the banks and crest of the cortex in a much wider distribution. And for the first time, the medial temporal lobe was involved in a diffuse pattern. And the pattern of pathology in the medial temporal lobe had similarities to diseases like Alzheimer's disease, but again, had laminar and topographic distinctions. This was stage 3, and then stage 4, sort of a widespread, more diffuse involvement affecting many, many parts of the brain, brainstem, diencephalon, even spinal cord. And we found that in the 35 or 36 former football players in this first series, that the stage of CTE correlated significantly with the age at death, and also with the number of years playing football, which we use as a proxy for cumulative repetitive head impacts. Several years later, then this was, the criteria were taken up by a NIH panel of expert neuropathologists, and the panel met to define the criteria, the neuropathological criteria for the diagnosis of CTE. And during that consensus meeting that we evaluated, I didn't evaluate, 25 cases of tauopathies were evaluated by 7 neuropathologists. They used the criteria I had proposed, and they did determine, they did identify all the CTE cases in that group, and they determined that CTE could be reliably distinguished from other tauopathies like Alzheimer's disease, PSP, argyrophilic grain disease, CBD, dementia complex of Guam, using this criteria. They also defined, and I think this was fairly an extraordinary event, that a pathognomonic lesion of CTE, meaning that if you see this lesion, you can be assured that the diagnosis is CTE. And the lesion that was defined was an abnormal perivascular accumulation of tau in neurons, astrocytes, and cell processes at the depths of the cortical sulci in an irregular pattern. Now, several years later, the consensus panel met again for a second time, and this paper is just coming into press. I think you'll see it next month in the Journal of Neuropathology, Experimental Neurology. In the second consensus meeting, 8 neuropathologists evaluated 27 cases of tauopathies, including 17 CTE cases that were varied by age at death. And they evaluated the successfulness of the criteria that were put forth by the first consensus. The goals of the meeting were to define the minimum criteria for diagnosis of CTE. They also wanted to determine whether CTE could be reliably distinguished from some age-related tauopathies, most notably RTAG, which had come into prominence over the past the few years preceding, and we needed to make sure that the criteria reliably distinguished CTE from RTAG, as well as primary age-related tauopathy, or PART. In this consensus meeting, we also looked at this restricted slide set of paraffin-embedded slides to determine if we could reliably stage the pathology according to the McKee staging scheme. So the results of the second consensus were that the NINDS criteria for the neuropathological diagnosis of CTE held up quite well, and the panel was able to identify all the cases of CTE. They also determined that a single pathognomonic lesion in the cortex was sufficient for diagnosis. They upheld the definition of the pathognomonic lesion, but they made several refinements. For one thing, the perivascular tau aggregates needed to be in neurons and neuronal profiles. They could have tau in astrocytes as well, but neurons had to be involved. So that was a distinction from the first consensus. They also agreed that subpial thorn-shaped astrocytes or perivascular purely astrocytic pathology represented RTAG and did not meet criteria for CTE. In addition, when they applied the McKee staging system to the restricted sample of slides, they found that it was inconsistent. So instead, the panel proposed an algorithm for categorizing, for evaluating cases for potential CTE, as well as categorizing it into low or mild CTE and high or severe CTE. So this was the algorithm the panel came up with. What you can see is it depends on a presence of a pathognomonic lesion. If one is present, the diagnosis is CTE. If you're concerned that the case is CTE, but you didn't find a diagnostic lesion, they recommend resampling of at least 4 to 8 bilateral cortical sulci to determine if the resampled material has a pathological lesion. And then in the evaluation of the severity, you're supposed to look in areas of the brain that suggest the disease is spreading. So you're going to look for NFT in the gyral banks and crests. You're going to look for NFT in the superficial cortical laminae, the hippocampus, other structures of the medial temporal lobe, thalamus, mammillary body, and cerebellar dentate. And if 5 or more of those are present, the distinction is then high CTE, and if less than 5 are present, it remains low CTE. And then just, well, about 6 months ago, this was published about the McKee CTE staging scheme. We looked at the staging scheme and provided a very detailed examination of the regional distribution of tau in CTE. We looked at 366 individuals that had been neuropathologically diagnosed with CTE, and we examined the association between CTE stage and semi-quantitative assessments of AT8 pathology in 14 brain regions. These semi-quantitative assessments were all done at the time that the case was diagnosed, so this was from a very large database. And we also did quantitative evaluations of AT8 pathology using digital scans and a perio digital quantitation. We also looked at age at death and dementia status and years of football play. This is just a demonstration of the digital scanner. And it turned out that the stages of CTE significantly correlated with the semi-quantitative ratings of tau across all 12 brain regions. They also statistically significantly correlated with the quantitative assessments of tau across the 7 brain regions. And you can see a representative section here from dorsolateral superior cortex. We found that the CTE-related pathology followed an age-dependent progression. Here you can see stage 1, stage 2, stage 3, stage 4. There is an age-dependent progression, but I would draw your attention to some of the mild stages of CTE and individuals in their 80s and 90s who are affected. Certainly, we believe most cases are clinically progressive. About 67% appear to be clinically progressive. Most of the cases appear to also be pathologically progressive, but there are cases, we would estimate 20% of cases that seem to either not progress or to progress very slowly. And this is just a heat map of the semi-quantitative pathology across multiple brain regions. We also looked at dementia status and found that the higher CTE stage was associated with increased odds for having dementia, which remained after controlling for vascular and neurodegenerative comorbidities. And replicating our previous work, we found that the stages of CTE correlated with years of play. And last but not least, we'd also did this something called a K-medoid cluster analysis. We determined that there were 5 clusters of pathology, 1, 2, 3, 4, 5, and they all correlated with age at death, dementia, and years of play. And cluster 1 correlated with stage 1, cluster 2 with stage 2, cluster 3 with stage 3, and stage 4, 2 very slightly different clusters with differing in the amount of tau pathology in various regions of the brain. So these results supported the usefulness of the McKee staging scheme, and they additionally demonstrated the distinctive regional distribution of tau pathology and CTE, as well as the links to age, dementia, and years of play. Now, why do we think tau is deposited in those regions after trauma? And you know, we always go back to the physics of the injury, the biomechanics of the injury, with the shear strain and stress being concentrated at crevices in the brain, like the cortical sulci, as well as at the interface of 2 structures of different viscoelastic properties, like blood vessel and brain parenchyma. This is a 3D computational model of helmeted head impact injury that accurately predicts the location of the CTE foci in the crevices, the depths of the sulci of the brain, and it reflects also the areas of the brain that have the greatest trauma-induced tissue strain. This was recently expanded upon in the Journal of Biomechanics, published just recently, where parts of the same team reconstructed 148 head impact events. They separated players into 3 different position profiles and showed that there were distinct differences in strain and strain rate across the whole brain between the players at different positions. There was a consistent spatial pattern of higher strain and strain rate in sulci compared to gyri in players at different positions. And I think interestingly, the largest sulcal strain and strain rate was in position 1, which was the defensive backs, the quarterbacks, and the wide receivers. Players in this position were exposed to the largest linear and rotational accelerations and velocity. And lastly, in this paper, they showed that there was a strong correlation between rotational velocity and acceleration and strain rate. So that rotational injury is really critical in the development of strain. And then this is from PNAS recently, where they looked at another finite element model of mechanical injuries. In this particular paper, they created a model of a single sulcus, as well as a perivascular region around a single vessel, and found that both of these regions were areas of the brain with high mechanical deformation during TBI. And of course, this correlates very well with where we see NFT in CTE. In addition, in this same paper, they did primary hippocampal culture on a elastic membrane with stretch injury. And without going into it in detail, they showed there was the stretch injury produced a mislocation of tau from the cell body to the dendrites and into the dendritic spines. And they could also show that there was synaptic dysfunction that was tau dependent in this paper. As for the symptoms of CTE, this is a paper from Stern et al. Back in 2013, where he noted that the symptoms, there tends to be 2 subsets of clinical presentation. There's a behavioral and mood presentation, which tends to occur in younger players, and there's a cognitive presentation that tends to occur in older players. In this paper in 2013, the age at onset for behavioral mood was about 35 years. There were only about 36 football players in that paper, with the cognitive onset about 59. As the players aged in either subset, they developed the symptoms of the other subset. So by the time the individuals died, they usually had both cognitive as well as behavioral and mood. And then these two subsets were later supported by our larger paper, our clinical pathologic case series that was published in JAMA, showing 177 American football players. There too, we found a behavioral and mood group with a median age at death of 66, and a second cognitive group with a median age death of 76. So there's still evidence of two subsets of onset as well as a course, and this continues to be somewhat mysterious. We're not entirely sure what the distinctions are between these two groups. This is from a paper, a large paper, I think an important paper that Phil Montenegro, a doctoral student in the group, put together. He looked at 202 cases of CTE that had been verified at pathology and looked for the most common clinical features in CTE. And this is what they are: memory, executive dysfunction, a lot of behavioral changes like violence, explosivity, a loss of control, and mood changes, which are often depression, but can be hopelessness and suicidality. He also described a progressive course in about 2/3 of the cases, and that a number of the cases had a long latent period between time of exposure and clinical manifestation. Now, there are some aspects of the clinical symptoms of CTE that I want to make very clear, because these are continually misrepresented. So I want to make this clear. All the clinical features of CTE might not be accounted for by the Tau pathology. The Tau pathology explains dementia, but it may not explain the behavioral mood disorders or many of the other aspects of the clinical course. There are multiple RHI-associated pathologies, and they probably all play a role in symptom production. We know that RHI is associated with changes in the white matter, loss of myelin, loss of axons. We know it's associated with diffuse inflammation, not just after RHI, but progressive increase in inflammation in CTE. We know that RHI induces a breach of the blood-brain barrier, loss of integrity of the small blood vessels. These are undoubtedly associated with some of these clinical changes. And we also know that RHI changes and accelerates and alters the deposition of multiple neurodegenerative proteins, including A-beta, including TDP-43, including alpha-synuclein. And these alternative pathways, the ones that aren't CTE and Tau, become increasingly important with age after RHI. So the older the person survives, the more these multiple neurodegenerative pathways come into play. And I'll expand on that in a little bit. The clinical features are nonspecific. There are no validated or widely accepted clinical diagnostic criteria, and there are no in vivo biomarkers that accurately detect underlying CTE pathology. Right now, it's a diagnosis of neuropathology. Advances have been considerable in understanding what the spectrum of clinical symptoms are in CTE, and that's largely been made through very structured, comprehensive, retrospective interviews with informants of individuals who were diagnosed at postmortem with CTE. It's also occurred in studies with participants who are at high risk for CTE, such as former NFL players. And as of right now, the current state of the literature regarding the clinical diagnostic criteria for CTE, they have been proposed for research, and they are known as TES, or traumatic encephalopathy syndrome. Now, research on the validity of TES is ongoing. It's not ready to be adopted into the clinic. And I can tell you that I think in the next few months, you will see advances about the definition and the criteria for TES. They are in the works. And then I wanted to just show you this paper that created a big mess or a big impact, certainly in the media world, but it also impacted our work quite a deal. You'll notice that the last 3 years of very high brain acquisition followed the JAMA paper. So this is the paper that was a large clinical pathological case series of American football players, and we found that 177 of 202 had the neuropathological diagnosis of CTE, including 110 of 111 NFL players, 48 of 53 college players, and 3 of 14 high school players. So very shocking statistics if you look at them at face value. But of course, this is a brain donation study. There's enormous selection bias that goes into a brain donation study. That was very clearly elucidated in this paper. Nonetheless, we were criticized quite profoundly by implying that this was the prevalence of CTE in the general population, and at no time did we do that. This was never our intent. This was strictly a clinical pathological case series. Subsequent to this paper, Biney and Bashinsky looked at, they estimated the prevalence of death of CTE amongst former professional NFL players by looking at all the NFL players who had died during that 8-year period where we had collected brains into the brain bank. And if we assume that during that 8-year period, we collected every single former NFL player that had CTE, that is, we got 100% of them, and they came in for brain donation, then the prevalence in the NFL players is 9.6%, about 10%. But if we assume the much more likely scenario that we only got 20% of the NFL players with CTE, or maybe 10%, both of these, I think, more reasonable estimates, then the prevalence of CTE amongst NFL players rises precipitously. At any rate, it's somewhere in between these two extremes. Then just last year, Jesse Mez did a really beautiful study where he accounted for all the just extremes of selection bias in the brain bank. And he did a very sophisticated study using simulation and inverse probability weighting in conjunction with our biostatistician, Yorgos Tripodis, and accounting for really extremes of selection bias. He found that the duration of play risk for CTE relationship remained consistent. In fact, what he found was that amongst these 266 former American football players, for every 2.6 years of football they played, their odds of developing CTE doubled. And if they played less than 4.5 years, they were 10 times less likely to develop CTE than those who played longer. And if they played 14.5 years, they were 10 times more likely to develop CTE than those who played less. So if you ask former NFL players, or you ask active NFL players like this reporter did, he asked Tom Brady, what about CTE? And Tom's answer was, first of all, I've been doing this a long time. So your body gets used to the hits. The brain understands the position you're putting your body into. My brain is wired for contact. I would say in some ways it's become callous to some of the hits. And so this is what it is. We have, to me, this is wishful thinking. It's denial of facts. But, I guess in the present, present day, this shouldn't surprise me. I mean, we've got so much denial of climate change. We had so many political issues centering on abnormal facts. So, this exists in CTE as well. Just a brief, I'm getting to 3:45, so you let me know if you want me to stop. I was just going to show you Aaron Hernandez, one of the players that came into our bank. He's been the subject of multiple books and documentaries, a Netflix documentary. And I wanted to show you his pathology because this young man, had a tragic life characterized by just many horrific events. But his legacy lives on. His brain has been really enormously useful in our research. We got the brain, it came into the brain bank. This is Victor Alvarez, one of my colleagues. It looked entirely normal when we received it. Nothing on the external surface suggested any issue, but when we cut into it, we could see ventricular enlargement immediately. There was atrophy of the fornix, and really shockingly, there was septal perforations. There's this degree of gross abnormality, very unusual in young individuals with CTE. In fact, when we went back and looked at our whole brain bank experience, we can't evaluate the septum in some individuals because we cut down the middle to freeze and fix. But in the cases that we were able to evaluate the septum, we found that approximately 30% of CTE cases have septal fenestrations, but we'd never seen it in an individual under the age of 40 until we saw it in Aaron Hernandez. I'm just going to show you that he had widespread CTE lesions. They were really classic. There were multiple. They were really riddled through the brain. Quite a substantial frontal lobe involvement, really almost half of the frontal lobe with Tau pathology. We evaluated him as stage 3. When we looked at his CTE lesions, we could see there was very intense inflammatory responses within the CTE lesion. This is from Aaron's brain showing the microglial marker brain. So I just can't get to all this other stuff within the time frame. I do think this is an important paper by John Cherry and John Crary, again, talking about that evolution of the isoforms of tau in CTE. It's a mixed tauopathy, but 4R predominance in early stages. The 4R positive cells are neuronal in early stages. They become astrocytes develop in CTE. It's age dependent, usually about 60 to 65 years in age. And then there's a conversion of tau to a predominant 3R profile. So an interesting evolution. And we know there's a new, so anyway, I'm gonna skip to the end. I apologize for not getting through all of this stuff. And I am going to thank my collaborators, a lot of my staff. They work really hard to get this work done. Many of them are here. It requires a huge team to do this work, and I want to thank you.
Thank you very much, Anne. So now we're going to open the floor to questions, which are going to be actually typed into the Q&A. We have already maybe 4 or 5 questions. But since I'm moderating this, and if you allow me, I wanted to ask the first question. It's just it's more a neuropathology issue here. So the last NIH, the upcoming paper, you were talking about mainly neuronal tauopathy as being the hallmark of CTE, and along the distribution, perivascular depth of sulci. Can you just comment on, actually, I think Willie is on the call also, but the Willie Stewart's paper in the ACTA, where they're mostly saying that it might be more tau in the astrocytes, if I understood well their paper. Do you mind commenting on that, please?
Yeah, well, I think, so the paper that I presented, we looked at 99 different cases of CTE, and there was a very large age stratification from 17, I believe, to 90 something. And you're not going to see this evolution of pathology unless you can see the very early lesions in the young individuals and compare them to the older individuals. And as I understand the ARENA paper, they looked at a limited number of individuals. All of them were in the older age group. I believe the mean age was at least 60 or 70, when astrocytes are a part of the CTE lesion. That's an evolutionary change that we do not see in the younger individuals. So I think that may explain some of the differences in our results.
Thank you. So I'm just going to get to some of these questions since they're just pouring in. We have now about 10 of them, 11 now. So I'll just go at the base. So George Hishaw, he's asking, many of the symptoms associated with chronic traumatic encephalopathy are similar to those experienced as a part of post-concussive syndrome. Some studies seem to have blurred the line and when symptoms of CTE may develop after repeated concussions. Can you speak to this?
Well, I think the line is very blurry, and I think it can be very, very difficult to distinguish persistent post-concussion change from early change in CTE. And I think the only way to really identify it is if you see a progressive change, if you see a lack of static symptomatology, which you would expect with post-concussive. You wouldn't expect it to increase over time, whereas CTE is a slowly progressive disease that tends to evolve.
Thank you. So Bianca Bridget Rock is asking, CTE has been highly researched in American football players and boxers. What are your thoughts regarding the potential contribution of soccer gameplay with acquired CTE or other trauma-induced neurodegenerative diseases? She's asking, especially since soccer is played by over 250 million individuals worldwide.
Right. Well, there are some early studies. Willie Stewart's published on some soccer players who've developed CTE. We have individuals who've played soccer in the brain bank who've developed CTE. So certainly the play of soccer is a risk for CTE. We don't have the numbers that would be needed to compare it to football, but my guess would be that the risk is there, but that it's less than what we see in American football. There's certainly plenty of literature in active soccer players regarding heading of the ball, and even in the absence of concussion, imaging studies showing changes on DTI, neuropsych changes after a single season. So my feeling is that the heading in soccer may be related to detrimental effects in the brain, and may be the primary factor that could lead to CTE. Of course, it's all complicated. There's never any easy answer. Heading is also associated with more collisions, player to player, more. But if we could eliminate heading in soccer, I think we would see a reduction in the risk.
Thank you. So Linda Van Eldlik is asking, have you seen any cases of CTE in female athletes, and does the pathology differ from males?
Well, this is a fabulous question. I wish I knew the answer. We are actively recruiting female athletes. We've had endorsements from, the world soccer champions, all sorts of really highly regarded female athletes. But the number of female athletes that have come into the bank has been just woefully small. Part of that is related to the fact that Title IX, it takes time for people to develop CTE, and also to die and donate their brain. We have not seen it in a female athlete. Do I think it could exist in female athletes? Absolutely. I think the concussion literature suggests that females actually may be more prone to some deleterious effects of repetitive head impacts. But I have no data on female athletes. All of the females with CTE in the bank, which are very few, were domestic or interpersonal violence.
Thank you. So I'm going to go to this one, which is going to touch other researchers who use, models, animal models, to look at concussion. So Andrew Greenhalgh, I hope I'm saying right his name. So he says, incredible work, thank you. To investigate mechanisms, do you think rodent models lacking sulci can ever recapitulate CTE pathology? And if not, perhaps the deeper regions such as locus coeruleus?
Yeah, I'm not sure. I think there's some very interesting results in rats who suffer repetitive mild injury. They don't have sulci. It's been obviously a real lack. I mean, I would say that the absence of a robust animal model for CTE, which you think we would be able to model, since we think it's trauma related, But I do think that it's proved quite problematic, and to date I have not seen a robust model of CTE in a rodent.
Thank you. So Ina Warner, sort of, I guess, basing it on this prion hypothesis of tauopathies, I guess, she's asking, well, she's first saying, amazing story, and thank you. And is there any evidence that neuronal tau as neuronal marker is taken up by astrocytes?
Again, I think it's a fascinating question. How does the tau get in the astrocytes? Does it come from a neuronal tau? I don't have the answer, but I think it's extremely interesting. But I unfortunately can't tell you.
Okay.
Not aware of any work on that.
Ian Berkey is asking, you briefly touched on the development of focal perivascular lesions as dependent on local brain mechanics. Do acute CTE pathology from the perspective of local brain mechanics increase the likelihood of future accelerated CTE lesioning? So I guess they want to say, if you have one, does that sort of spread around? I'm not sure. That's my interpretation.
I'm not really sure either. We definitely see perivascular abnormalities in young individuals with CTE, like Aaron Hernandez. He had tremendous numbers of hemosiderin-laden macrophages in his white matter surrounding blood vessels. We're doing some intensive vascular studies now with Clarity and other things to really look at those early vascular changes in CTE. Could it drive the pathology? I think, yes, it could, because the breach of the blood-brain barrier are allowing molecules that are normally excluded in, driving inflammation. I think it could set up a real feed-forward process where the damage to the blood-brain barrier causes inflammation, inflammation causes tau, and it cycles forward.
There is a question from Georg Hischau. He's saying that not all players with repeated injuries would develop CTE. I think that sort of stems from your work also. Some theories of Alzheimer's are that individuals with problems in repair process. So now I'm not sure what type of repair processes they're talking about that lead to, so there are issues in AD with repair processes, and that leads to development of pathology. Do you have any thoughts of similarities between these Alzheimer theories and those who develop CTE?
Well, I didn't get to that part of the talk, and I feel badly because obviously not everyone gets CTE. People with the same type of exposure, the same dose of exposure, some people get it, some people don't. It's not absolute. We think that the genetics are extremely important. We're just getting to the point where we can point to some genetic susceptibility and resilience genes, but you know, I think you have to stay tuned, because I do think there's a genetic a component to this. We know that ApoE4, it's not a risk factor like it is for it's not a risk factor for CTE like it is in AD, but it is associated with worse tau severity, worse dementia. We know that the team MEM106B allele that acts as a resilience gene. There's less inflammation, a greater synaptic density, and less tau deposition with individuals with that particular allele. We're looking at the MAPT haplotypes. There's a story there, but the folks that are doing it aren't ready for me to talk about it. And then there's things like race. I think we're going to see race coming out as a differential in the future. We're going to be looking at age. Well, age is an instigator in this pathology, like it is in Alzheimer's and almost all neurodegenerative disease. But then there's things like occupational attainment, educational attainment, exposures to others, environmental pathogens, pesticides. I mean, you know, there's a big, big list. And I think that it's going to be a very complex interaction of multiple factors in any given one individual.
Michael Zainen is asking, would be great to hear what you wanted to say in more detail about RHI and risk of other neurodegenerative disorders. Well,
I can tell you quite quickly that RHI in and of itself, 8 years of playing a contact sport, and we did this work, this is Torstein's work in the Framingham heart study subjects. We found that 8 years or more of playing a contact sport increased your risk for Lewy body disease by 6 times. And that was in the absence of CTE. These are not individuals who develop CTE. So RHI accelerates alpha-synuclein deposition. We're looking now at a larger cohort to see if it's Parkinson's or if it's just Lewy body disease. We know that TDP-43 is deposited in about 50% of the brains with CTE, especially those in the later stages. But RHI, is it an accelerant for TDP-43 pathology? I think the jury's not out on that, or is still out on that, but I think it may be. A-beta is accelerated by a history of repetitive head injury. It's deposited at an earlier age and at a faster rate, and it contributes in the presence of CTE to the presence of dementia and functional impairment. So I think there are multiple ways the brain can go wrong, and RHI may precipitate all of them.
Thank you, Anne. We're at the top of the hour. I think we're going to stop here. There are still plenty of questions. I'm not sure if you have access to them, or we can answer them online maybe later. So anyways, I thank you for your time.